thank you so much for this really nice introduction. So it's really great to be here. It's great after a long time being associated to TUM, now being really member of TUM since 1st of June. And I'm really happy to tell you a little bit about these fancy small little creatures called microorganisms and how they influence soil quality, environmental services, and subsequently also human health. And I think this is something what you all know, that since a very long time, it is very well known that if you want to be a good farmer, you have to know more about your soil. And taking into account that the microbial biomass in the soil is a major part of this ecosystem, I think it is also worth thinking about the microbiota that live in the soil and what they do and how they can be influenced. When I talk about microbiota, I just want to show you some of the pictures because microbiota is far more than bacteria. Sometimes people just say this is bacteria and this is the same as microbiota, but there are far more. When we talk about microbiota, it's fairly everything smaller than 50 micrometers. So everything what you cannot see by eye can be considered as microorganisms. And when you look under the microscope, you can really see this is a fascinating world, and you can see the protists here, you can see the fungi here, there are nematodes around, there are bacteria, of course, around, but there are also the archaea, which are very important drivers also in soil ecosystems and which trigger, for example, nitrogen cycle and carbon cycle in the soil. So it's a whole bunch of different organisms, and it is not only the bacteria. However, mainly in the last century, people were thinking that these organisms, let's say, are quite unsocial. So they just reproduce themselves and they might be interacting with their environment, but there is no interaction between the microbiota themselves. So the famous postulates of Koch from 1896 say, when you want to know what a microorganism is doing, you have to isolate the microorganism, you have to bring it back into the system, and then you have to confirm that there the functionality that you were proposing is really gained. And this has been mainly done in the context of diseases, but later on, also people, for example, isolated rhizobia, they isolated mycorrhiza, they isolated a lot of environmental bacteria, they brought them back in the particular ecosystem, and they could confirm that these microorganisms are really important and they provide important functionalities also for the soil ecosystem. However, today we know that these microorganisms are not unsocial at all. They build very complex communities, and just when you go out in autumn and you look on litter degradation, you see a large number of different microorganisms that are interacting to degrade the litter. There are those microbes which do the shredding, there are those which degrade the lignin, there are those which provide the nitrogen and the phosphorus. So there's really a huge interaction of different microorganisms, and this is what we call microbial communities or the microbiome. And when you just look here on the scale here of a lignin material, you can see that it's not only one particular group of microorganisms that is involved in this degradation of this lignin material. You can see you need very different microbiota that this lignin material is degraded. You need those which really do the action of the degradation of the lignin, but then subsequently you need those microbes which provide the nitrogen. You need those which provide the phosphorus, and those microorganisms, of course, benefit from those microbes which degrade the lignin because they are provided then with the carbon. So it's really an interaction of very different functional groups of microbiota. And when you then try to calculate something like networks, what we love to do in microbiology, you can see that there's a very strong interaction of these organisms. And you can guess when you take one of these organisms out, the complete network structure will change. And subsequently, also the process of litter degradation will be different compared to the situation where you have this particular network structure. You can even do it on a molecular level, and you can see then the interaction of the very different processes that are integrated into the litter degradation. And I think this is very important, and it shows nicely that one particular microorganism is not able to do all the steps that are needed for litter degradation. This is really an interaction and a share of work of very different microorganisms, which then contribute to the process of the litter and the lignin degradation in those cases. So I think we have to revise the postulates from Koch a little bit, and we have to say that beyond these single microbiota, which of course are very important to study and which are very important also to isolate, we also have the microbiomes, and this is the interaction of different microorganisms. And when we want to understand the functionality and the resilience of ecosystems, we have to look far more on the microbiomes and their interaction than on a single microorganism who is involved in one particular process. And this leads to the definition of a microbiome. So it's the microorganisms on the one hand, like I told you, the bacteria, the archaea, the fungi, the protists, the algae, the nematodes, and most likely many more. But what they do is they provide a so-called theater of activities. And this is what we are interested in when we talk about soil quality. We want to know the turnover processes. We want to know how these microorganisms trigger plant growth, how they are involved in carbon sequestration. And this is the theater of activities which we are very much interested in. So it's the microbiota and their activity. And of course, what is unique with microorganisms is that they can adapt to changes in the environment. And their phages, mobile genetic elements, and so on play a very important role. For plants and higher eukaryotes, it's not so easy to take up DNA from the environment, integrate it in the genome, and then really express the functionality. For bacteria and other microbiota, it is a very important trait to adapt to certain environmental conditions. Then they take up genes from other organisms and they express them in their genome, and this helps the organisms to adapt. So it's very important that we also understand this adaptation of microorganisms to changes in the environment. And of course, like I told you, microorganisms are interacting. So it is very important to understand the signal molecules. So how do the microorganisms communicate with each other? And subsequently, how do the microorganisms and the microbiome communicate with host systems? How do microorganisms communicate with plants? How is our gut microbiome communicating with our gut epithelium? This is very important, and therefore we have to understand, of course, also these signal molecules, and on the other hand, of course, also molecules which then interfere with this interaction of microorganisms or of microbiota with their corresponding host system. And this led us to the definition of what is a microbiome. So it, as I said, it's far more than looking on bacteria only. And microbiology in general is a science direction which is very much driven by methodological developments. I told you microorganisms are always very small, so you cannot see them by eye. So you need advances in technologies to see these microorganisms and to isolate them and to get their functionality enhanced. So the first major step was, of course, the development and the identification of the microscope, which then enabled researchers really to see these really small creatures. And subsequently, of course, there were a lot of different methodological developments which helped us to understand these different microbiota. And now also in the last years, this new sequencing approaches helped us also to unravel the diversity of the microbiota that we have in soil ecosystems, that we have in our human gut or in any other environment. And therefore, I think we have to keep in mind that each methodological advance that we make helps us to get more information about microbiota, their functionality, but also their adaptation to particular environmental conditions. Microbes can colonize any environment on this planet. I think this is very important. So we find microorganisms not only in soil and in marine and freshwater ecosystems, we also find them in very extreme environments, like, for example, hot tubs. We find them on glaciers. We find them in the desert, so they can adapt to these very dry conditions which we have in these particular environments. And this flexibility really helped the microbiota a lot to adapt to these very differing ecosystems. And a microbial alive on this planet without microbiota would not be possible, which you can see also from this slide, where you can see where microbiota play a very important role for us when we only think about the food production system. Where are the different steps which could be done without any microbiota? Fairly, there is no step starting from the growth of the crops until the processing and the final production, for example, of bread and so on. Without having microbiota, this would fairly not be possible. And there's a very nice article. I really recommend this article to read. It's from Jack Gilbert and Josh Neufeld, and they asked the question, is there a life without microbiota possible on this planet? And they started with the discussion saying, oh, all these microbiota are pathogens and we would be happy if these microbiota are not around. And it was a situation like here in the restaurant, and they looked at their beer and their wine and they said, oh shit, when we don't have the microbiota around, we wouldn't have wine and beer. And with this, they really started this discussion, is a gnotobiotic life, a life without microbiota possible? And at the end, of course, they came to the conclusion, a life without microbiota on this planet would not be possible, because they fulfill so many different ecosystem services for us humans, for the environment, for all ecosystems, that without them, this planet would not be able to live. And you can see, and I think this is very important also when you think about the sustainability concept of TUM, where One Health plays a very important role, that these different microbiomes are interacting. So it is not like you have the human microbiome, you have the plant microbiome, you have the soil microbiome, you have the waste microbiome. No, all these microbiomes are strongly interacting. So when you change the plant microbiome, subsequently you have changes in the soil microbiome, and this also feeds back then to the human microbiome via bioaerosols or via food and feed quality. So this is really interacting. And I think this is very important to realize that any change you make here on the human side, or any change you make here on the plant side or on the aquaculture side, for example, this feeds back to all the other parts of our ecosystems. And this really helps us a lot to understand what is really behind this One Health concept. And I just want to remind you here that this concept says that in general, when you have a healthy environment, this promotes human health and animal health. In contrast, when you have an environment that is not healthy, this will lead on the long run, at least, to also problems with human health and animal health. And when you see all these environmental diseases that are occurring at the moment, you realize that this concept is really true, because we have significant problems in the environment resulting from the climate change, from the biodiversity crisis. And this feeds back to human health and also to animal health. So when we want to prevent diseases, I think the best thing would be to start in the environment and to think to how we could improve environmental quality. And you can see here a very nice showcase where this is really then conceptualized, where we try in a project to improve food production, to produce healthy food, and to see how this then interact also with human health Of course, there are a lot of different parameters which in addition influence human health. So this is not a direct path. When you have healthy food, automatically humans will be healthy. But it shows that the food production and the food quality is a very important driver of human health. So when we improve food production, make it more sustainable, automatically we might improve also our personal health. And I think this should be a very strong motivation to think about how we can improve food production systems and food quality subsequently. But when we talk now about soils, are there microbes? How many microbes there are? And it is fascinating when you go to the soil, you can consider soils as a hotspot for biodiversity. So these numbers are really fantastic. And when you talk to people and say in 1 gram soil, there are living more microorganisms than human on Earth, this is really a number that shows you that these microbiota are indeed very important. And it's not only the pure number, it's also the number of different species that you find there. So in 1 gram of soil, you might find 10 to the 5 different species. And when you bring it more on the level of strains and ecotypes, it might be even far more. And this is what is in this very small amount of soil available. And it is, of course, the question, what are these microorganisms doing? Do we need all of them? Do we need only a very small part of them? Can we substitute them once they might be getting off a particular system? This is all questions what is science at the moment all about. But just to remind you, when we talk about soils, we talk about ecosystem services that is provided by these ecosystems. And most of these ecosystem services are triggered by microbial activities. So when we talk about food production, it is obvious that microbiota play a very important role for that. They provide the nutrients for the plants, they protect the plants from pathogens, they produce phytohormones, and there are many, many other features that microorganisms are doing. When we talk about the filtering and the buffering of a particular soil, where we want to safeguard our groundwater, so the degradation of all these pollutants is done by microbiota. When we talk about carbon sequestration, storage of carbon as a mitigation for climate change, it is microbiota which do the initial processes, and vice versa. There are also microorganisms around which can promote climate change by producing methane, by producing N2O, and all these climate relevant trace gases. So it is very important to know under which conditions a soil is a sink for carbon and under which conditions it might become a source of carbon. And microbiota, of course, play a very important role for that. But also when we speak about things like erosion protection, which is a very important issue in the Alps, for example, microbiota play a very important role because they can produce something like a glue which then helps the soil to stick together and which reduces the erosion capacities. So for all or for almost all of these ecosystem services, microbiota play a very important role. So I think it is justified to have a more deeper look into microbial communities and ecosystem services provided by soils. And then when we talk about the drivers for microbial communities, so which factors influence the microbiota? It is on the one hand, of course, very site-specific conditions. It's the soil type. It's the content of sand, clay, and silt. Consider you have a very sandy soil. There's a lot of oxygen available. So you get a lot of microorganisms which use the oxygen. When you have a soil which is under very clay, there is not so much oxygen available. So microorganisms are in favor, which can use alternative electron acceptors, like the nitrate, for example. And they do denitrification. There are other site-specific factors like the soil pH, for example. The temperature on a particular site is very important. The water content that you have in the soil. Imagine you have only very low water content. There's only very few specific microbiota which really can cope with these particular conditions. Also, the other animals around, the plants around, play a very important role in shaping the microbiome of a particular soil. And then, of course, it is agricultural management when we go now to the production side. It's the tillage, for example, which strongly influences microbial community. It's the amount of fertilizer. It's the quality of the fertilizer. So it is a difference if you apply organic fertilizer, which first needs to be transformed by the microbiota into ammonia and nitrate, or if you apply inorganic fertilizer, where already ammonia and nitrate is available. This all triggers microbial communities. And I think it is very important that it is not one factor which influences the microbiota. So this would be easy when we just say it's the pH, which is the main trigger. This would be easy because then you can just manipulate the pH in the direction you would love to have your microbial communities. It's a bunch of different factors, and this makes it so difficult to steer and to manage microbial communities in a particular soil ecosystem. And you can see here the major drivers for microbial diversity losses in soil. And that's, of course, the high amounts of nutrients that are available in the soil. So those microbes which are really involved in the degradation of dead biomass, they are reduced more and more because simply there is too much ammonia and nitrate available. It's also the pollution, for example, which is a very important driver for microbial communities and which reduces also community structure in soil ecosystems, because you can imagine that these pollutants do not only affect humans negatively, but also some microbiota. It's the management and mainly monocultures strongly reduce microbial diversity in the soil, because you repeatedly grow the same crop there and you get a very strong enrichment of particular microorganisms, and all the other ones are dying off. And by this, you get a reduction in the structure of a microbial community, diversity losses, and any loss in diversity is associated with a loss of functionality. So when you lose, for example, microorganisms which are able to fix nitrogen, it is obvious that legumes, for example, cannot grow on a particular site. When you lose, for example, Arbuscula mycorrhiza, it is obvious that many plants cannot be grown on a particular site because they need this interaction with their particular communities. So in any case where you have a loss in microbial community structure, you also have a loss in microbial community functionality. And then this is ongoing, this changes and biodiversity losses. This is not only true for the macro world, where this is very well documented. This is also true for the microbial world. And this is experiments that we are doing in the frame of the biodiversity exploratories, where we investigate how land use intensity influences microbial community structure. And you can see here for the bacteria, but also for the fungi, the higher you have the land use intensity, the lower is the diversity. So there's a very strong correlation between microbial community structure and land use intensity. And more important, it is not only a complete loss in biodiversity, you also change these networks that I was referring to at the beginning. The morphology of these networks, though the overall functionality of these microbiomes changes quite dramatically. And of course, not surprisingly, on the long run, plants get problems to grow and you have to intensify your management more and more. Plants are less and less becoming resistant towards stressors. So you have to supplement this reduced functionality by either adding chemicals, by either adding high amounts of fertilizers. And I think this is very important, that these losses in biodiversity, every farmer can see in a very short run, because he has to do more and more management and has to apply more and more chemicals that plant growth is really occurring. And it is not only plant growth as one of these functionalities and ecosystem services I was referring to, it's many of these ecosystem services that I was referring to are very much influenced by these changes in diversity and by shifts in land use intensity. And this is something which is not only true for agricultural soils, it's also true for forest soils. Though this pattern that when you have a reduced diversity, you also have a reduced functionality and a reduced multifunctionality of these particular ecosystems is true, independent if this is a typical agricultural soil or a particular forest soil. So the problem of modern agriculture, I think, is very obvious. We have the need to produce high quality crops. We have the need to have high yields. And this is why the farmers use inorganic fertilizers. They use pesticides. They use very reduced cropping sequences. This leads to reduced carbon stocks in the soil. And subsequently, you get a reduction in biodiversity, and you have to apply more fertilizers. You have to apply more pesticides. So this always gets more negative, negative, negative. And this also impacts, of course, at the end, also food quality. It impacts also our climate. It also impacts many other ecosystem services. And of course, also the quality of neighboring environments, either the groundwater or non-managed natural environments. And all of this is accelerated, of course, by climate change scenarios, mainly when you consider extreme events like drought. So what we want to have is we want to turn this around. So we need approaches now which are referred to something like smart agriculture, where we want to increase microbial diversity in the soil and their activity, where we want to increase carbon stocks again, where we want to have efficient nutrient cycles where the nutrients are in the system and are not pushed out of the system, either by leaching or by N2O emissions, for example. And this requires low input agriculture. It requires low levels of mostly organic fertilizer. It requires the use of reduced pesticides, reduced soil management, and increased cropping sequences. So coming back from this 3-part cropping sequences, for example, to 7 parts in the cropping sequence. And again, this would have positive effects for food quality, climate, ecosystem services. But the key for that is really smart agriculture. So what could be smart agriculture? In our eyes, it could be, for example, that you use new amendments, where you really bring back diversity to a particular ecosystem that is low in diversity. You could use also precision agriculture, where you just give the amount of nutrients according to the needs on a particular plot and not apply 200 kilograms of N per hectare when it's not even used. You can use biofertilizers, bioinocular, where you know that these microorganisms support plants in their growth performance and in their responses to stress. And of course, you can use new forms of cropping, like intercropping, which is a very nice system to increase also biodiversity, because you have different plants, you have different roots, you have different exudates coming from the roots. And by these different exudation patterns you have in such systems, you increase biodiversity. So this is something where I think we can really start to improve smart agriculture. And by having these approaches, we automatically come into these positive feedback loops by having increased carbon stocks, all of that having positive quality effects for food quality and subsequently also for the complete ecosystem. And this is something where I showed you already or explained you already that the microbiota associated to the plants are really very important. And this is why we try to understand how the plant recruits its microbiota from the soil. And of course, when you think about recruiting, if something is not present, you cannot recruit it. So it's the question, what type of microorganisms does a plant need for recruitment? And are these microorganisms present? And if not, can we substitute them to a particular soil ecosystem to improve crop quality and also crop yield. And I was talking about important features for the for the plants already, which are related to nutrient degradation. The biocontrol of phytopathogens is very important. The production of plant growth promoting factors, hormones, the so-called plant probiotics. We have the mycorrhiza, which is very important for the transport of nutrients for the increasing of a rooting area where the plant then really can take up nutrients and water. Of course, also degradation of pollutants is very important, but also UV protection when we think about leaves is very important for the plant to optimize its growth and to become really persistent against certain different stressors. And when we talk about leaves, one very important functionality that is not mentioned on that slide, but which becomes more and more important, is that microbiotas produce volatile organic carbon substances. And these substances are very important for attracting insects. So when these substances are not produced, the plant might not be able to attract insects. And this, of course, has consequences then for next generation of the plants and also for the yield and for the performance of a particular plant in the generation itself. So these leaf microbiota become more and more important. Many studies were just focusing on the root microbiome, but now more and more studies also focus on the leaf microbiome because people say this is really becoming more and more important. So what are the acquisition strategies of a particular plant? So I told you on the first glance, when a seed starts to germinate and there are small roots occurring, the first recruitment comes from the soil. So there are microbiota around and the plant gives carbon to the soil and this attracts the microbiota. And by attracting the microbiota, they get enriched in the root area and they provide their functionality. Then at later stages, of course, above ground, there's also very important input coming by the air and by bioaerosols. So also there, the plant tries to recruit those microorganisms that are really needed. But then there is something missing. And I think this is very important to know that there is also the seed. And in the seeds, there are microbiota which are transferred from generation to generation. So there is a vertical transfer from generation to generation of microbiota. So this cycle here is very much closed. It's very similar to the human cycle or to any cycle of higher eukaryotes, where you on a first glance have vertical transmission. Think about the mother provides the first microbiome to the baby, and then subsequently the baby starts to recruit its own microbiome, which is then depending on the type of diet, which is depending on the environment. And this is exactly what the plant is doing. It gets its first microbiome by the seeds, and then it starts to recruit microorganisms from the soil and from the air. And think about when you have soils with a low diversity, or when you have a system where the air is strongly polluted and you don't have the right microorganisms in the air, then this recruitment is not possible. And then the functionality which is provided by those microorganisms is no longer existing, which means the plant has difficulties in its particular ecosystem, yields are reduced, quality is reduced, and stress response is also strongly reduced. So what we are doing at the moment is we try to work on several aspects of improving this type of agriculture. We think that breeding is a very important issue. Think that the plant provides carbon for the microbiota which live in the soil. Think about the old races where you have a huge microbiome and a huge diversity in the root system, just because there's a large amount of carbon available and which is deriving from photosynthesis. For the modern cultivars, most of the fixed carbon goes in above ground biomass, not much is going into the root. So there is not much microbiota around, and so we think that there needs to be some crossing between old races and modern cultivars to ensure that these microorganisms which are needed for the plants are really in the rhizosphere. We are also working on the cropping sequences, on an intercropping. I told you that we think that this has very beneficial qualities also for the microbiota in the soil. We have new biostimulants. I will show you some of them in a minute. And we are, of course, working on management approaches like applying high carbon amendments, for example, to the soil, because this would mean then that the microorganisms are forced, for example, to mobilize also nitrogen and phosphorus, and this would then also be beneficial for the plant. So for all of these approaches, microbiota play a key role, but as a microbiologist, of course, I like this approach with biostimulants most, because this is then something where you really can understand also the functionality. And we have a large number in the last decades identify of novel microorganisms from soils, from plant surfaces, where we think that these microbiota might be very beneficial when we inoculate them in the soil ecosystem. It's bacteria where we have a very strong focus on, but of course, as I told you, microbiota are more than bacteria. There are also a lot of fungi around, mainly mycorrhizal fungi, that have been isolated and where it has been shown that it is really beneficial to apply these microbiota to the soil. And this is not only fiction. There is a lot of preparates and systems on the market which you can really buy, and where also not only private persons in their garden apply those bacteria, but which also large agricultural consortia apply these particular bacteria, because it is well accepted that these bacteria really provide help to the plant and improve plant quality and plant yield. So what we think is, and just to remind you to the first pictures that I showed you about this microbiome and the interaction, having only one microorganism inoculated might not be enough. These microorganisms might need certain other microorganisms around. So what we try to develop is something like microbial consortia, which we inoculate. And for that, we first try to get all the isolates in hand. There are now consortia available, for example, for barley, where 4,000 isolates and more are available. And then we try to mix them in certain ratios with certain functionalities. And we see if this then helps to make this bioinoculation business more sustainable. Because at the moment, it is really, you have to apply them each season again, because these organisms do not survive in the soil when you introduce them. We think when you apply them as a consortium with different functionality, as a small microbiome, like I showed you at the beginning, then it might be able for these organisms to shape their own niches, and by this also to survive in the particular soil ecosystem. And just one example, which I really like very much, this is apple replant disease, where when you repeatedly grow apple on a particular field, you can see that after 3 or 4 generations, apple plants are no longer growing. This is true for all Rosaceae, but for apple, the economic burden is the highest. So this is the question, what happened in this soil? And if certain microbiota can help to compensate for these problems that are occurring there. And we could identify with a molecular approach microorganisms that are able to degrade phytoalexins. So these are toxins that are produced by the roots to avoid that pathogens really attack the plant. And obviously these phytoalexins accumulate in the soil. And this leads then to a misinterpretation of the apple plant after some generations. And we could identify certain microorganisms which are able to degrade these phytoalexins. And we thought it might be a good idea to use those microorganisms as bioinocular. But then we also realized that maybe these plants also need some more microorganisms which protect them from attacks by phytopathogens. So we thought it's good to complement these bacteria here, which degrade the phytoalexins, by bacteria that prevent the plants from attacks by phytopathogens, so biocontrol active organisms. And finally, we also identified some apulscular mycorrhiza, where we thought, wow, this might be important for nutrient uptake. And we mixed these three bacteria together. And finally, we could really show also now in field trials that this really helps to mitigate when we are talking about this apple replant disease. Another approach that we are trying derives also somehow from medicine. I think you all know something like stool transplants, where people have an infection with Clostridium difficile, and then you get a transplant of the fecal microbiota from a healthy person, and without knowing which are the microorganisms, but in 90% it helps, because then the Clostridium difficile is outcompeted of the gut, and the person becomes healthy again. And we thought, wow, this might be a cool approach to do very similar things also from the soil. So why not taking soil from an environment which is high in biodiversity and substituting it to a soil which is low in biodiversity? And we are trying this now together with Horsch and colleagues in Brazil, where this approach really is becoming more and more important. And obviously it worked. At least for 2 generations now of plants, we could show on a large scale of 160 hectares that the amount of fungicides needed is strongly reduced, just because we introduced important microbiota from this healthy, high biodiversity soil to this degraded soil. And this really helped then for biocontrol of the plants growing on this particular site. And this is a very similar concept. We don't know which are the microbiota which are really active and which do the job, but we do not care. We just take the soil, apply them here. And we think that this might be also an approach without having in-depth knowledge about microbiota. Take a soil from close by, which is the same soil type, which grows under the same environmental conditions. Take the soil and apply it to a degenerated site and see if this works. And first experiments we are doing now together with the colleagues also in Germany, where we want to show if, or we want to prove if this is really working. So we published, as it's always very difficult to convince people about something which they do not see. And this is the problem with microbiota, you do not see them. So we published a paper where we really showed success stories for the system where really microorganisms were introduced into the ecosystem. So just to convince people that this is not fiction, but this is really true, what we are telling. And there is another very important paper where we really say it is important to maintain microbial diversity. It is important, we have to keep microbial diversity. It's as important as biodiversity on higher trophic levels. We have to maintain the biodiversity. And when we reduce biodiversity in any ecosystem, either freshwater, marine, soil, whatever ecosystem, it reduces the functionality of these ecosystems. And this is a consensus which was done by, I don't know how many people were involved in writing this particular article. So I think this is very important. And when we go a step further, and I think this goes now back to the first slide that I was showing you, there's really a call that we should keep and protect microorganisms in any ecosystem to maintain global health, and not only on the level of microbiomes, but we really also have to know which are the microorganisms behind that. I was talking a lot about microbiomes, but of course, at a certain point, we also want to know which are the microbiotas which do the functionality, like I showed you with this reconstruction of these synthetic communities for the improvement of plant growth. This is something I think which is very important, that we really try to protect and find means how to protect microbial diversity in soil ecosystems. And this again, of course, is something which I think is very important. And this is why I love this double role at the Helmholtz Center and at the university. I think it is not only important that we find therapies for certain diseases, that we develop new drugs, which I think is very important to help people who are sick, But on the other hand, I think we have to prevent diseases. We have to find means how to prevent diseases, and their environmental quality and improvement of environmental quality, not only in landscapes around cities, but also in cities in itself, play a very important role. And I think this is nice things that we are doing here also in the frame of the graduate school, where we really try to improve microbial diversity, but also diversity of animals and of plants in the urban environment. Also in the project with Wolfgang Weiser, we are doing very similar things. And I think this is very important when we speak about prevention of diseases. We have to bring biodiversity back to urban environments where many people live. And I think we have cool concepts now developed from the agricultural ecosystems. And we have to see if we can apply these concepts that we have now also to urban environment. I forgot to mention Monica, of course, with her urban farming systems, which also go in a very similar direction. So this is, I think, something very important and where we really want to contribute also in the next years with that. But of course, also food production and agriculture is a very important focal point when we speak about prevention of diseases. So with this, I think I'm through. This is Hippocrates, who already postulated also 3,000 years ago that there's something like a One Health concept. When you have healthy environments, you have healthy humans. When you have environments which are not healthy, humans get sick. And this is, we call it now One Health concept, but Hippocrates already postulated that 3,000 years ago. And with this, I think I'm through with my presentation. Most likely I was far too long, sorry for that. But people told me already that there were colleagues speaking much longer, so I was not the longest one. Thanks for that.